So let's get started. The topic today is toxicity. And since I obviously can't here talk about all of toxicology, what I really want to restrict this lecture to is toxicity with respect to small molecule drug discovery and drug development. And when you're talking about toxicity, what you really need to consider is that toxicity can either be acute or chronic. And the main reason why we care about toxicity is that we want to prevent something of still doing more Windows configuration. Does anybody have a laptop with the slides? This is embarrassing. Okay guys, let's simply go on. I just keep talking with the slides up here. So the reason why we of course care for toxicity is because we want to prevent some things like thalidomide from ever happening again. Before thalidomide was taken off the market, it had caused more than 10,000 birth defects. You all have the scary images. Children were born without arms or legs or without both. And here's the culprit, here's thalidomide. Very simple molecule. And the tragedy was really that people thought that because it's very similar to an amino acid, it would have very little toxicity. Our thalidomide, you see the chiral atom here, had the desired sedative effects, as thalidomide was teratogenic. And really everything went wrong with this molecule that could go wrong. As thalidomide was only teratogenic in humans, monkeys, and rabbits, and not in mice and rats where it had been tested. And you'll see later that this led to a drastic change in the Food, Drug, and Cosmetic Act in 1962. Ever since thalidomide, toxicity now has to be evaluated in 2 species, one of which is not a rodent. So nowadays, before you can give a drug to patients in phase 1 trials, it has to be tested for toxicity in, for example, rats and dogs or mice and monkeys to prevent something like this from happening ever again. And of course, very few drugs are used in pregnant women. This is a central agency that regulates drug development in the United States, the FDA, the Food and Drug Administration. This is a very powerful agency. It regulates food, drugs, medical devices, vaccines, blood products, and cosmetics in the United States. It has the power to approve, restrict, and withdraw drugs from human and veterinary use. But it also has duties. The FDA has to inspect the factories of facilities that are producing food and that are producing drugs. Do you know what these abbreviations GLP and GMP mean?
Good laboratory, good manufacturing processes.
Exactly. And this is what the FDA will inspect. So for example, the FDA will inspect the factories in India and China that are producing active pharmaceutical ingredients and make sure that they're really only manufacturing one compound in one room so that there's no cross-contamination. They want to see all the records of the starting materials. Then when the API is shipped to Ireland, and this is a very realistic scenario, where the tablets are pressed, there needs to be a full record of what went into the tablet, where did the starch come from, where did the lubricating agent for the tableting machine come from, how much went into it, and how controlled was the whole process. So that when a pharmacist here in Davis dispenses a tablet, you can really trace back where everything in this tablet came from, in case something happens. And the FDA also oversees the preclinical and clinical trial system. However, this has not always been so regular. A little more than 100 years ago, there were basically no rules at all. This was literally the Wild West. But this was a time when all the big cities were growing, when people were moving off the land and starting to consume more and more preserved and tinned food. And this gentleman here, Harvey Wiley, who was a chemist, wondered if that was safe. So he founded the so-called Poison Squad, which was a group of young men, mostly chemists, who would eat capsules with what was used as preservatives with their food. Borax, salicylic acid, formaldehyde, saccharin, sodium benzoate, and copper salts. Since the formaldehyde made these young men violently sick, it was banned as a preservative, and Americans became interested in food safety, which until then really wasn't a topic. Another thing that was in the air at this time was his book, The Jungle, from Upton Sinclair. He had actually intended to expose the exploitation of workers in this book, but because he was describing practices in the meatpacking industry, and there were shocking descriptions in there from workers falling into the rendering tanks and being ground up with the meat product, the public was simply shocked. And foreign sales of American beef fell by 50%. And together, this started to build support for a federal agency. And actually, Harvey Wiley, and with support from Roosevelt, was really the founder of the FDA. So in 1906, we have the Pure Food and Drugs Act, which created the FDA. For foods, it prohibited the alteration and misbranding of food. It introduced meat inspection. And for drugs, it required labels that described the content and declared whether they contained alcohol or heroin. And at that time, it was perfectly okay for your drug to contain this, as long as you declared it on the label. And these were drugs that were used at the beginning of the last century. Mrs. Winslow's soothing syrup, 65 milligram morphine. You know, you use a little too much of this and your pesky infant never cries again. Seriously, this was a time where you had a lot of unexplained child deaths because people would use something like this. It was also a common practice to take the pacifier and dip it in tincturia opium and give it to the infant. Then he would sleep through the night. Cocaine toothache drops, 15 cents. Or here, heroin hydrochloride, the cheapest specific for the relief of cough. Initially, this was really ironic. Heroin was made because people thought it would be less addictive than morphine. And then, of course, there were things like this, the electromagnetic bathing fluid. This is real patent medicine. This was supposed to cure everything. Scarlet fever, epilepsy, mercurial eruptions, whatever that might be, cholera, rheumatism. I mean, this is a real fraud. But, you know, these are very, very potent things. And then things kind of muddled along, and you'll see that this is a common theme. You always need some disaster for the law to change. And in 1937, we had the first sulfonamide antibiotic. Sulfanilamide was a very safe antibiotic. And it was pretty much the first time that infections could be treated. Unfortunately, somebody had the idea to dissolve it in diethylene glycol, add raspberry flavor to make it sweet for children, and sell it basically as an elixir. More than 100 children died from kidney failure because of the diethylene glycol. This is antifreeze. And this, of course, again led to a public outcry. And in 1938, we have the Federal Food, Drug and Cosmetic Act, which now required pre-market safety approval for all new drugs. We still didn't have to show that it was effective, but it had to show that your drug product was safe. It prohibited false therapeutic claims, so the really obscure patent medicines vanished. It introduced more food standards, and it brought cosmetics and medical devices, which until then were completely unregulated, under the jurisdiction of the FDA. The major change in the 50s was the introduction of prescription, because it was no longer felt appropriate that people could just go into a pharmacy and demand whatever drugs they felt like. There are certain drugs where it was felt that you needed a physician to treat the disease. And then, of course, we have thalidomide. What I didn't tell you before was that these birth defects only happened in Europe and in Canada. Thalidomide was never sold in the United States. And the reason was this lady, Frances Kelsey, who was a reviewer at the FDA and who had concerns. She wanted to see more safety data, so she blocked the approval of thalidomide in the US. So in 1962, we have the Kefauver-Harris Amendment that really changed the game. They now require safety and efficacy testing for all new drugs. And the safety testing included testing in 2 species for toxicity, one of which is not a rodent. These amendments require GMP and later GLP. They started to control prescription drug advertising. They introduced patient protection, because there had also been some abuses in clinical trials where patients didn't know that they were on a clinical trial. And, very important for drugs, it required the retrospective review of drugs that had been approved before 1960. So at this point in time, many old drugs were taken off the market, and it was also reconsidered what was safe enough for over-the-counter. So this really cleaned up the landscape. And with these amendments, we now have the path to drug development as it is in place today. We have a preclinical phase where the target is identified. You discover a lead molecule that engages this target. You optimize it. You test it for toxicity and metabolism. You study animal PK. You look at animal efficacy and the disease model. Then all this data is combined into a so-called IND, Investigational New Drug Application. The FDA has 30 days to look at this. If they don't protest, you're free to start testing your drug in phase 1. Phase 1 is basically safety and pharmacokinetics in a few healthy volunteers. And you're primarily looking at the pharmacokinetics, because remember, we talked about species differences. You never quite know how different the PK will be when you go from animals to humans. Phase 2 is the first efficacy testing in people with the disease. You get a first idea of whether your drug might work. And then you do the real deal. The large, statistically powered, double-blind, placebo-controlled clinical trial. You know what double-blind is?
Yeah.
Okay. And then all this data is combined and submitted to the FDA again as a new drug application. On average, this takes 12 to 15 years nowadays and costs $800 million to $1.5 billion to really bring a drug to market for a bigger indication. And what is always disheartening here, this is the likelihood of your drug reaching the market. And you see here, in the preclinical phase, you have a very low chance of your drug reaching the market. You only have a pretty decent chance after a successful phase 2. And I probably should down-correct this number here, because recently the FDA has been denying a couple of drugs here after phase 3, because they felt they were not safe enough. But let me give you a real-life example. This is a drug that was developed by a friend of mine, zichronotide. This is a cone snail toxin. It was found to block calcium channels in 1984. They demonstrated that it had efficacy in animals. You see, the numbers are difficult to see from here. So in 1980 it was discovered. In 1984 they found that it blocked calcium channels. In 1989 they demonstrated that this compound treated pain in animals. My friend then left the university and started working with Elan Pharmaceuticals to develop this. In 1993 they had the phase 1 data demonstrating that this is safe in people. In 1995, phase 2. In 1998, phase 3. In 1999, they submitted this to the FDA. The FDA took a year to review this and came back in 2000 and said, guys, this is approvable, but we don't like your statistics in the phase 3 study. Please redo that. If you're a small company, that is a big problem. So they totally regrouped, spent a lot of time raising more money, redid the phase 3 study, resubmitted the new drug application in 2003, got approval in 2004, at the end of 2004, and finally launched the drug in 2005. This is a new drug application. This is the amount of data that was submitted to the FDA. And I think now you understand why the FDA takes a year to review this. And this is my friend when he started working on the drug, and this is when the drug reached the market. So, I don't think I really need to make a comment on this. Drug development takes a long time. And it costs a lot of money. So the question is, why do people bother to do it? You can make a lot of money with drugs. These are the global sales for pharmaceuticals between June 2012 and June 2013. $854 billion. And about half of this is in the United States. These are the top 10 selling drugs in the world. You don't need to really learn all of them, but it's a good idea to have at least some idea about what are the most common diseases and what drugs are really the so-called blockbuster drugs. Number 1 here is an asthma drug. Number 2, Humira. This is anti-TNF for rheumatoid arthritis and psoriasis. A statin for high cholesterol. Another anti-TNF reagent, a drug for acid reflux, a small molecule. Another anti-TNF reagent, Abilify, a small molecule for depression. Insulin Lantus, slow-release insulin for type 1 and type 2 diabetes. Zymbolta for depression, a small molecule. And anti-CD20 for non-Hodgkin's lymphoma. You see, 5 of these top 10 selling drugs are biologics. This is a little skewed by the fact that many of these drugs have very high prices. Many of these drugs run around $20,000 to $30,000 per year per patient. So what is always good to look at the same time are the most prescribed drugs in the US. And interestingly, the most prescribed drug is hydrocodone. This is Vicodin for pain. And we could spend a lot of time how much of this is real need and how much of this is abuse. We have a statin, we have an antihypertensive, thyroid hormone replacement for hypothyroidism, another antihypertensive, the racemic version of Nexium, omeprazole, 2 antibiotics, metformin for type 2 diabetes, and hypochlorothiazide, a diuretic also used as an antihypertensive. So together, these 2 lists are always pretty informative because they give you an idea of what are the most common diseases in our society. But you might have also heard about this. I used to say the patent cliff is approaching. By now the patent cliff has happened. Many of these so-called blockbuster drugs, this term means a drug that is selling for more than $1 billion, have come off patent. So they have become available as generics. What has come off patent recently are Lipitor, Zeroquel and Clarix. Lipitor, for about 6 years running, was selling for around $13 billion. This has eroded the income of the pharmaceutical companies, and that's why we've seen this major restructuring. These mergers between Pfizer and Wyeth, Merck and Schering-Plough, and restructuring in their research and development. I always find this shocking. Between 2000 and 2013, the pharmaceutical industry has cut 300,000 jobs and closed some major sites. Not only Sandwich in the UK, this was Pfizer's original site, but also closer to home, for example, Roche has closed its Palo Alto site. And we've seen some massive outsourcing, especially of chemistry, to India and China. Although this is reversing at least a little bit more recently. Okay, I don't want to totally depress you, so let's come back to toxicity. So why do we have such a big attrition rate in drug development? If you analyze why drugs fail, you see that 30% fail for lack of efficacy. These are drugs that worked in the animal model, but then simply don't work in people. Because there's typically something about the human disease that we haven't really understood. 20% are business decisions. These are perfectly good drugs that get abandoned because management decides we're not going to work on antibiotics anymore. Or we're going to abandon our epilepsy drug in favor of the diabetes drug because we can't afford to develop both. But the large pie here, 44% fail for safety or toxicity issues. And when we're looking at toxicity, what you really have to differentiate is on-target toxicity. This is toxicity caused by engaging the primary target. For example, TRPV1 is a heat-sensitive ion channel. Blocking it worked wonderful for pain in animals, but if you gave TRP1 blockers to people and to primates, you raised the body temperature, you gave them fever. And since this was caused by the primary target, this is nothing that you can get rid of through structure activity. This kills this target. And then we have off-target toxicity. When your drug, in addition to the primary target, is hitting something that you didn't intend it to hit, like a cardiac sodium channel, or this cardiac potassium channel, which when you block it, always induces arrhythmias. But this is something that you can address through clever medicinal chemistry. You can try to dial out the side effect. Another reason for toxicity is the presence of a toxicophore. If your molecule contains a group that is intercalating, phototoxic, or alkylating, or if your molecule generates a toxic metabolite in liver metabolism. That is called indirect toxicity.
Question?
Yeah?
What do you mean by phototoxic?
I'll explain this in a little later, but phototoxic is something that is not toxic under normal light, but which becomes very toxic under UV light. I don't know, have you ever picked those plants with a really milky juice? There are certain plants, when you pick them and the juice runs over your skin and you go out into UV light, your skin starts to blister. That is phototoxicity.
Okay.
And I explain in a minute how that works. But let's start with the toxic metabolite. And probably the most famous toxic metabolite is the metabolite of acetaminophen, Tylenol. Who has taken Tylenol here in the room? Wow, okay. It's typically a very safe drug. Problem is, when you overdose it, it is actually responsible for 90% of the acute liver failures in this country. Here's Tylenol, acetaminophen. Very simple compound. You see it already has a hydroxy group. So typically it gets glucuronidated or sulfated and excreted. However, P450 is always around. I always tell the medical students it never sleeps. So it will also try to oxidize acetaminophen into its quinone. Normally, that's not a big issue. You get a little quinone. You've learned we have glutathione in the liver. Remember, that was our detoxifying agent. It provides SH groups. So this is a mitral acceptor. Glutathione will react with it and inactivate it. And remember, we have about 10 millimolar glutathione in our liver available for this reaction. However, if there isn't enough glutathione, what will increasingly happen is that you alkylate liver proteins. And this really becomes a numbers game. If you take more than 4 grams of acetaminophen per day, you're exhausting the available glutathione, and you get more and more covalent binding to liver proteins. And that is liver toxic. So like I said, acetaminophen is responsible for 90% of the acute liver failures in this country. So now you might ask, when are you overdosing acetaminophen? There's 2 situations. One is suicidal intent. These are typically teenagers. And I shouldn't give you suicide advice, but Tylenol is really the worst thing you can pick for it. Because if you take enough, you die a miserable death of liver failure for a couple of days. If you get saved, you might have damaged your liver so much that you need a liver transplant. So both is not a nice scenario. The other group of people who tends to overdose Tylenol are elderly people with pain who can't control their pain and who keep taking more and more and more. Because they don't realize that if they have rheumatoid arthritis, there's actually something better to treat it than Tylenol. So if you see your grandmother taking another and another Tylenol, you might want to talk with her about it. And actually, because this keeps happening, that people destroy their liver with acetaminophen, the FDA has just required that there is a red warning label on Tylenol. It now says contains acetaminophen. Read the label. Because as I said, if you take the normal dose, it's a perfectly safe drug, very well tolerated. If you overdose it, this is when you get the liver problems. And we'll know in a couple of years if this has any effect. Okay, here we have another famous toxic metabolite. Halosane was widely used inhalation anesthetic, but in a small group of people, it induced an immune-mediated hepatitis. This was again P450 generating toxic metabolites. In this case, 2E1 is reducing halosane. It's first oxidizing it to this unstable halohydrine, which is then eliminating HBr. You get an acid chloride here, very reactive, and again, it's alkylating liver proteins. Halosane can also be reduced. It's reduced, eliminates bromide. You get a radical, which is reactive. You can reduce this radical further. You get a carb anion, which is eliminating chloride, and you get a very reactive carbene, which is actually then suicide inhibiting the P450 enzyme. That is called suicide inhibition if P450 is generating a metabolite that is so reactive that it immediately reacts with the active site of the enzyme. And then what happens in a few predisposed individuals is that the immune system picks up these modified proteins and makes an immune response. Same happened here with this diuretic. This diuretic would get oxidized by 2C9 to a sulfoxide. And again, this is a mitral acceptor. And it can react with liver proteins, and the immune system can pick out these modified liver proteins. So I think you're getting the idea that there are certain structures that are toxic. And that's why they're called toxicophores. We have frank electrophiles, molecules that do not require metabolic activation and that can directly react with proteins, such as alkyl halides, mitral acceptors, reactive haplocycles, which are set up for nucleophilic substitution, or epicytes. Also cross-linking agents, anything pyrrhous disulfides that can make disulfide bridges, metal chelators, imidazoles, triazoles, and pyridines can complex the iron in P450. And then the phototoxic and DNA damaging compounds like cypsoralates. And I'll come back to those in a second. Then we have toxicophores that require metabolic activation. Alkynes can generate epoxides, this is P450 oxidation. P450 can take aminothiazoles and generate these reactive light ketones. You've already seen this with anilines. They can be oxidized to quinones. Benzodiazoles can generate carbene. And even benzodiazepines can be oxidized to these mitral acceptor-like structures. Some more here. Hydrazines can be oxidized to azo compounds. Furans and thiophanes, you've seen this with thionylic acid, can generate epoxides or sulfoxides. Or methyl indoles can be oxidized to these mitral acceptor-like structures. Nitroaromatics can become nitroso compounds and generate reactive oxygen species. And also para-hydroquinones can be oxidized to quinones and generate reactive oxygen species. So let me explain the psoralates and the phototoxicity. All these plants that burn the skin typically contain these type of compounds. They have two reactive double bonds, one here, one here. Have you guys had any photochemistry at some point? Or are you totally blank on photochemistry?
Very, very light.
Okay. There are certain structures that are good at generating triplets. And most of photochemistry is triplet chemistry. So these type of double bonds can be excited easily. And then they undergo 2+2 cycloadditions with other molecules that have double bonds in the right configuration. And what is really nasty about the psoralates is that they're flat, so they fit into the DNA, so they can intercalate like this. And then when you shine UV light on this, you get a 2+2 cycloaddition with the DNA bases here and here, and you cross-link the DNA. Very nasty. They can also react with lipids. And what is really the most acute toxicity is they can generate singlet oxygen in the skin. And I don't want to go too deep into photochemistry here, but psoralates, when they have the right configuration, can pass. I mean, this is what happens every time you excite something with UV light, you generate the singlet. But if you have the right electronic conformation, you can get an intersystem crossing and go into a long-lived triplet, which then can actually react with other stuff. Normally, you just excite into the singlet, it drops back, and you emit light. But if you excite into the triplet, you get a long-lived species. And in this case, the long-lived psoralane triplet will meet triplet oxygen. Oxygen is weird, it's normally existing in the triplet. So the psoralen triplet will give its energy to the triplet oxygen, and you get singlet oxygen. This is one of the most reactive oxygen species that exist. You turn singlet oxygen loose in a biological system, and it will oxidize cholesterol and unsaturated fatty acids. It will oxidize amino acids, and it will cross-link proteins and lipids. If you do this with these little brine shrimps, you put a psoralen into the bath, you shine some UV light on it, these little things turn brown within a couple of hours because you generate singlet oxygen. So in summary, we have frank electrophiles, structures that are cross-linking or that can directly react with proteins, DNA, lipids, and cause toxicity. These type of structures can typically be easily identified if you switch on your chemical brain. Just ask yourself, does this look reactive? Is there any position here that smells like an SN2 reaction? Or anything else reactive? Then we have other compounds that generate reactive metabolites, anions, cations, quinones, carbenes, during metabolism. These can, if they're very reactive, suicide inhibits the P450 enzyme while it's generating this metabolite. They can produce reactive oxygen species. This is called oxidative stress. And they can alkylate proteins. If you generate a lot of this metabolite, you get direct impairment of liver function, like with acetaminophen. If you don't generate a lot of this metabolite, you might trigger an immune response in predisposed individuals. And that is typically very difficult to predict. This is relatively easy to pick up in development. You also typically recognize these type of compounds relatively easily. But this is nasty. This is difficult to predict. And of course, there's a lot of prediction software out there, and in the interest of time, I don't want to belabor this. What this prediction software is basically doing, it's doing the same like you do with a toxicophore list. It's checking the molecules for toxicophores. There is software out there supposedly predicting carcinogenicity or hurt liability. Most people will look at the software and then do the experiment anyway. But these type of softwares are useful for generating alerts. But what you would typically do when you test a drug preclinically, and this is, for example, what Bristol Myers Squibb does, but most companies will do something similar. You test for cytotoxicity, you test for teratogenicity, you do a screen on a panel of important receptors and transporters and enzymes and see if you're hitting anything that you don't want to hit. You test for homologs of your target. So if you're developing a kinase inhibitor, it's most likely to inhibit other kinases. So you test for selectivity over kinases. If you're developing an ion channel blocker, you test for selectivity over other ion channels, because those are your most likely off targets. And then you look at hepatocellular toxicity. And after you've done this, in general, you want to take a good look at your structure and ask yourself, is there anything toxicophoric in here? But if you step back and look at this list, furans, benzofurans, thiazole, there are thiophene. Many of these structures are so-called privileged structures, which are likely to be good drugs. So it's a very difficult decision if you really combine something that might generate a toxic metabolite without really testing for it. So every company has some slightly different policies here of what they will pull for toxicity concerns. And let me finish again with a real-life example. This is a compound we're taking in phase 1 clinical trials. They're actually going on right now. It's a blocker of a voltage-gated K-channel in lymphocytes. And the first thing you see here, now after this lecture, is a big fat toxicophore. This is a psoralen. And I don't want to go into all the details why we chemically could not replace this psoralen, but we just couldn't. We would always lose activity. So at some point we decided, okay, let's test if this thing is really toxic. So we looked at selectivity over other K-channels. We looked at Ames test. We did a lead profiling screen over receptors and transporters. We had a bit of P450 inhibition here. But when we looked at what psoralens are really known for, cross-linking DNA and producing single oxygen, that was negative because with a side chain, this molecule is too big to go into DNA. And with this 5-position substituent, it doesn't produce triplets. And if you don't make triplets, you don't make single oxygen. So that's what I'm saying is when you're checking for toxicophores, use your chemical sense. And then just to briefly round this off, after you've done all this in vitro work, you go into animals. You give the compound at a relatively high dose for 28 days or 3 months to animals, and then you look at blood chemistry. You look at liver enzymes. If the stuff was liver toxic, liver enzymes would go up. You look at you look at hematology. If your compound was really toxic, it would kill white blood cells. And then you do the really nitty-gritty work. You sacrifice the animals, you section the tissues, and you have a really good pathologist go through all the sections and look at them. Because if something is liver toxic, you see this at how the liver looks like. Because you see dead hepatocytes, you will see changes. Same if something causes other defects. And there's really, so far, no replacement for really doing this. This is really the last step you do before you go into clinic. Thank you. It was interesting that no laptop connected.